Hello everybody, this is Dr. Lampert speaking. This lecture will be about not only fungi, but also about ecological interactions. And in fact, we'll start with the interactions. And the reason for the discussion of interactions is because I want to sprinkle these ecological concepts through the biodiversity, like I have been for communities and mentioning and how to study biodiversity, things like that. But the fungi are a great example because they can interact with other organisms in pretty much every conceivable way. So I went back to the fun pictures here. These are actually all different ways in which we interact with fungi. So I'll take a second, let you pause, kind of think about what kind of interactions we have with all these examples, and then I'll discuss them. Let's start with the top left. So the top left shows a trophic interaction. This is an interaction in which we eat a fungus, and a very specific fungus, and we start seeing things. We go on a little trip. We get blasted off to space talking with the coyote gods. So a lot of fungi use chemistry to protect themselves, and those chemicals can have some pretty remarkable pharmacological effects. So we'll talk about those more toward the end. Let's go counterclockwise. So another fungus that we eat is the mushroom. And here I took a nice photo of the Hardee's / Carl's Jr., depending on where you're from, mushroom Swiss burger. I remember in high school this was about the top of the line fast food. And this is an example of another example of us eating mushrooms. But in this case, there's no pharmacological effect other than feeling full. So we can also eat mushrooms as a source of nutrients. Okay, in the bottom right, John Mann's least favorite fungus, athlete's foot, but luckily there's boom, tough-acting tenactin, and we can cure it with medicine. So in this case, the interaction is the fungus being a pathogen of humans, and so we can get some pretty obnoxious diseases from fungi. And in fact, although we don't usually think of fungi killing a whole bunch of people, unless you play the Last of Us video game, but they can be pretty nasty pathogens of wildlife and agriculture. So fungal pathogens can have huge effects on people. Okay, and then we'll finish in the top right with an interaction. So this is an interaction in which we use fungi to get a product. Specifically, we use yeasts, which are a type of fungus, to get ethanol, and even a little bit of compounds associated with flavor too. So we associate with fungi in a lot of ways. We eat them for nutrition, we eat them for pharmacological effects, we use them to produce our food in several ways, and they can be pathogens of fungi. And by the way, I'm not saying these are the only ways we can interact with fungi, so I invite you again to pause and list any others that you can think of. So the learning goals are to, first of all, be good at interactions. So be able to look at interactions, be able to figure out what type of interaction it is, what's the name of an interaction between different species based on what the outcomes are. We'll do a little more life cycle. These life cycles might be a little more intuitive compared to plants, but there's one curveball that the fungi throw in there as far as life cycles are concerned. And then we'll do a little fungal classification. So pretty similar types of things to what we did in the plant concept. Okay, so for the reading, again, these sections are consistent whether you have the 9th or 10th edition of Campbell Biology. So chapter 31 is the fungus chapter. This has all the information your hearts will desire about fungal diversity and fungal life cycles and interactions with fungi. And then chapter 54, we're actually going to blast ahead because that's a nice section that defines interactions, things like predation, mutualisms. Those are all defined and described in Chapter 54, section Concept 54.1 in particular. So I definitely recommend you read all of those. We have a research article for this concept too. We may or may not cover it depending on where we are in class. We're going to have a handout for this concept about interactions, so we'll definitely be covering that depending on how we're doing in class. I'd like to do a reading exercise for this article as well. Alright, so let's start this concept by talking about interactions. So we have a lot of different creatures that interact. So we have prokaryotes, protists, and plants, which we've already covered. And fungi and animals are also in communities, and they'll be interacting with things. Now, remember how I when I first brought up communities, I said that it's several populations of several different species that that either interact or have the potential to interact. So it's pretty important to have a clear definition of interactions. And I'll give that as a social or trophic, in which case, which means eating, meeting between different organisms, either individuals of the same species or different species, and that will have an effect. That meeting will have some sort of effect. So the creatures are going to be acting on each other, and it's going to be an interaction because they're reciprocal actions. So one species' actions can affect the other species' actions, which can then in turn affect the first species' actions. So that's how we'll define interactions. And what's interesting about interactions too is that any interaction can affect other interactions. So what I mean by that is an interaction between a plant and a fungus might have an effect on an interaction between an herbivore and that plant. An interaction between a plant and an herbivore might have an effect on the carnivore. And these interactions can be direct or indirect as well, in which what that means is that an interaction between two different organisms can mean they either interact specifically with each other or that interaction is mediated by a third party, which happens quite a bit as well. We'll cover those all more when we talk about trophic ecology, however. Okay, here I have a way of looking at interactions. And we'll spend a little time in class to work on this, and then we'll do a handout for interactions as well. So interactions can be defined by what the outcome is for each of the actors. And so interactions can be positive, negative, or neutral. Neutral, of course, being neither positive nor negative. Interactions can be positive, negative, or neutral for both actors. And any actor can have any outcome. So an interaction that's positive for one actor could be positive for the other one, or negative for the other one, or neutral for the other one. So here I have kind of a simplified way to represent all these different interactions. So I have it separated by actor 1 and actor 2, and each of the cells is going to represent a different interaction. So the top left cell of this table would be an interaction that's positive for both actors. The top right would be negative for actor 1, positive for actor 2. Bottom left would be, bottom left cell of this table would be negative for actor 2, positive for actor 1. And the bottom right cell, of course, would be an interaction that's negative for both actors. So what I'll invite you to do here is spend a little time thinking about these and coming up with some examples of these interactions. Now we will discuss these in class briefly, and we will have a worksheet for these, but I'll invite you to kind of think of some now. So think of something that's positive for both. Think of something that's negative for both. Let's go back to what I covered on the introduction slide. So think of us eating mushrooms on a burger. Is that positive or negative for us? Is that positive or negative for the fungus? What if we have athlete's foot? Before it's treated, is that something that's positive or negative for us? Once we treat it, Is that an interaction that's negative or positive for the fungus? Has the outcome of that interaction changed for us? Is that now a positive or negative interaction for us? So think of lots of examples. In class, we'll think of broad categories. So what's a broad category of interactions that might be positive for both? What's a broad category of interaction that might be negative for both? So we'll cover those more in class. So an interesting question that I'll pose before we move on, or move away from the interactions and move more specifically into fungi. So think about a saprobe. So think about a fungus that is decomposing an animal. So where do you think that might fit in here? Where do you think saprobes might be on this table? Something that's interesting to keep in mind here. If one of the actors is dead, can there be an interaction based on the definition I gave? We'll talk about that a little more in class. Okay, let's talk about the classification of the fungi. So, we covered the land plants for a concept, which is just one group of archaeplastids. The fungi are a group of opisthocons, which is in the unicont super kingdom, the supergroup. And the uniconts are going, or the opisthocons, excuse me, are going to consist primarily of us, our closest relatives, the choanoflagellates, fungi, and their closest relatives, the nuclearids. The opisthocons are separate from the amoebozoans. As you can hopefully guess, they are not shaped like blobs. They are not little soup pod covered amoebas like all the amoebozoans are. So the opisthocons are the uniconts that are not amoeba-like. And the uniconts are actually named after the fact that they have flagellated gametes. And what's interesting is that most of the uniconts, except the animals, of course, a lot of the uniconts, the animals and the choanoflagellates have not lost their flagellae, but a lot of these other organisms have. But ancestrally, that's a shared characteristic. Let's start with yeasts first. We have to start with yeast before we start talking about classification, because yeast is a term like algae. Remember I said algae is a term for any aquatic or semi-aquatic plants or plant-like creatures. So yeast is a term for single-celled fungi. And the yeasts can be in two different divisions and some different classes within those divisions. So yeasts are actually not a monophyletic group. So there are yeasts are not based on shared ancestry. They're in two different divisions. So because of that, yeasts tends to be something that is not valid from a taxonomic standpoint. Like I can't say class yeasty or anything like that. Here's a nice diagram of yeasts. So showing their good old asexual budding that I'm sure a lot of you guys have heard about. So that's how they reproduce, just by producing small daughter cells as a result of mitosis. Mostly asexual, but not all. Alright, so the rest of fungi, however, are going to have mycelia. And that is the body, quote-unquote, of a fungus. So a mycelium is a network of thin filaments. And all these filaments are connected to each other, and they can exchange nutrients and other solutes between them. And the threads themselves, the filaments that make up the mycelium, plural, hyphae, individual, hypha. And hyphae are going to be the cellular structures as well. So the hyphae might be split into individual hyphal cells. We call those septate hyphae because septum is going to separate individual cells of the hypha. Sometimes hyphae might be cenocytic, as in contrast, and cenocytic, as you could probably imagine, are hyphae that do not have septae, and so all the cells are connected to each other, and you can't really differentiate the cells in that hypha. So they're kind of opposite terms. So if we recognize that fungi can be grouped into either yeasts or large mycelial fungi with large mycelial networks, then the next question is, well, what does a fungus do? So a fungus doesn't really move. A fungus is like a plant in that regard. It doesn't move at a rate that we can really see it. It doesn't move the way we generally define moving. But they do grow into things. So they grow through something called a substrate. Substrate is just a generic term for the surface that something sits on or can grow within. So fungi grow into a substrate. They secrete enzymes to digest the substrate. So it's going to be, for the most part, an organic substrate. They will grow into the substrate and digest it, and then they will absorb the nutrients from the substrate into the mycelium. So, if that's how a fungus works, then how does the fungus get its nutrients? So think of it this way. Can a fungus be a producer? Can it be a saprobe? Can it be a predator? Can it be a parasite? Can it be another type of symbiont besides a parasite? So think about these. So when I have Tile 1107, I'll very often define the idea of producer and consumer roughly the same as autotroph and heterotroph. And then I will often present a question in the class asking if fungi are producers or consumers. And in general, fungi are always consumers. Now, there is kind of a wishy-washy exception to the rule of fungi being consumers, although technically the fungal part is a consumer, a lichen. So we'll cover that in a little while, but a lichen is going to be a producer, or at the very least a mixotroph as well, or instead. So fungi can sort of be producers if you consider a lichen a fungus. Mycologists tend to study them. Lichens are in the fungus chapter of the book. A fungus is a part of a lichen, but not the whole thing. But we do see that fungi can be all of these other things. Can a fungus be a saprobe? Absolutely. So the fungi that we eat are going to be saprobes. The fungi that you see in dead trees are going to be saprobes. Let's save the predator one for last. Can a fungi be a parasite or pathogen? Of course they can. So if the organic substrate is alive, the fungus is going to be a parasite or pathogen. If the organic substrate is dead, a fungus is a saprobe. And what's interesting is that some fungi don't really make a distinction. So some fungi are parasites of something while it's alive, and then once it dies, they just keep on feeding off it, and it becomes saprobes. So we see this very, very often with parasitic fungi or pathogenic fungi that result in death of the host. They just keep feeding on it after it's dead, although they will quickly send spores in the environment to infect fresh live hosts. Fungi can actually be other types of symbionts besides parasites and pathogens. They can be mutualistic symbionts, which is a type of symbiosis that's beneficial to both to all the actors in the symbiosis. Now, what's really interesting is predators. So predators is probably the least likely of the consumer lifestyles. But it turns out that fungi can even be predators. So they're so versatile in the way that they interact, they can even be predators. So they can actually feed on microbes. in a predatory way. So I know when I talked about protists, I was saying all wishy-washy different ways to be a predator of a microbe, and our predators of microbes, real predators. Fungi can attack nematodes, microscopic animals. They can attack other microbes, and they can kill and consume small individuals. So because of that, fungi can even be predators. So even though the major lifestyle of a fungus is to grow into an organic substrate and digest it, they can sometimes be predators of microbes that share that substrate with them. The last bit of basic biology of fungi that we'll talk about before I talk about the fungal groups is the life cycle of a fungus. And there's something very unique and distinctive about a life cycle of a fungus that makes it worth knowing. So they, like all living things, they can have asexual and sexual reproduction. So everything from bacteria to animals has some asexual reproduction. So most animals are sexual, but there are some animal species that can reproduce asexually. In the case of asexual reproduction, it's pretty straightforward. So spores are haploid. Spores are always haploid. Spores will germinate and grow using mitosis into haploid mycelium, and then at some point that haploid mycelium will produce a sporangium to produce spores, all through mitosis. So that is an important thing to keep in mind, by the way, is that asexually fungi can produce spores through mitosis, whereas plants always produce spores through meiosis. So do be careful to keep that in mind. The sexual reproduction, the sexual part of life cycle of fungi is a little more interesting, and what's interesting about it is that the way that fungal cells come together. So plants and animals, and in general protists, have gametes. So they have single haploid reproductive cells that come together to make a diploid zygote. In the case of fungi, they generally don't have gametes. What they have are hyphae that merge, and it's actually a two-step process. So I'm going to call this merging. I'm going to call that fertilization. So it's actually a two-step process, and there's a step called plasmogamy, and that is the joining of hyphae, or cells of the hyphae, to each other. What's interesting about plasmogamy is that the contents of those cells don't combine. In particular, the nuclei don't combine. So the true definition of fertilization, in which nuclei come together, that doesn't happen right away in a lot of fungi, most fungi for that matter. Instead, we get what we call heterokaryotic cells, and that's just a term for cells that do their thing, divide, and all that, with different nuclei inside. So hopefully you all remember hetero means different, and kary means nucleus. So heterokaryotic cells have different nuclei in them from different individuals. And at some point in these cells, karyogamy does occur, and so the nuclei do come together right when the fungi are reproducing. In general, as part of sporogenesis, the heterokaryotic cells will merge, or the heterokaryotic nuclei, I should say, will merge in a process called karyogamy. So kary again meaning nucleus. Gamy means to join. So you hear gamete, plasmogamy, karyogamy. So gam means to join in biology. And then the zygote goes through meiosis to produce spores. So that's a little different from us and from plants, right? Because the zygote in animals and plants, and some protists for that matter, will produce an embryo which is going to develop into an individual. In the case of fungi, it's really the spores that develop into individuals, that develop into mycelia. So the zygote's going to go through meiosis pretty quickly. So a couple different things going on here in fungus sex. So a little bit different than us. Not quite as complicated as plants, though. So let's look at the groups of fungi. So here is the phylogenetic tree of fungi with a list of all the numbers of species, and so we'll cover each of these different groups. Notice that they all have hyphae. They all can grow into some sort of substrate, some sort of organic substrate. Something that I will point out to you, if you look at this tree, you have these branches leading to the chytrids and zygomycetes. You see these weird little three branches coming toward them, or three-line branches coming toward them, whereas the glomerulomycetes, ascomycetes, and basidiomycetes all have a single line. So the chytrids and zygomycetes are interesting because they're not monophyletic. So they have multiple branches of multiple lineages leading to the taxi that we recognize today. So that's kind of an interesting thing here. By the way, if you were to draw characters on these trees, the chytrids actually don't have heterokaryotic stages, but other fungi do. Let's talk about each of the groups specifically. The chytrids. So they're essentially the ancestors of fungi. They're the closest thing to a opisthokonch common ancestor. They are not very derived. They do have hyphae. What's interesting about them is they still have flagellated spores. If you remember, I mentioned opisthokonch refers to being flagellated. So they're the group that retains flagellated spores. And because they have flagellated spores, they need to live in an aquatic environment. So they tend to be aquatic saprobes, or in some cases pathogens. So they're kind of like oomycetes in that they can kind of blur the line between saprobe and pathogen depending on if the organic substrate is alive. And it turns out pathogens is where we hear most about chytrids. We'll cover that a little bit later when we talk about pathogens. Outside of the chytrids, most other fungi are named after the structures that produce spores, or spores are dropped out of. So I'm going to introduce each of these other divisions of fungi, zygomycetes, ascomycetes, and basidiomycetes. I'm going to introduce them all through the context of their reproductive structure. So let's start with the zygomycetes. So first of all, you'll notice that we see the same term over and over: zygomycetes, zygosporangia, zygospores. So zygomycetes are named because they have something called a zygosporangium. Sporangium is the spore-producing vessel, the spore-producing structure. Hopefully you all remember the term sporangium from class, or from a concept about plants. So zygo, let's see, where have you heard that term before in class? So you might remember heterozygous. You might just remember zygote. So zygosporangium, it's the same word root. It means to come together. So what happens is when plasmogamy occurs, the sporangium, will form out of the area where those hyphae from two different individuals comes together. So the sporangium forms where two different hyphae from different individual fungi comes together. So that's where the zygo comes from. And of course, the spores are also named after the sporangium. They're named after their zygospore, or so their name zygospore. Okay, the ascomycetes and basidiomycetes both have something called a sporocarp. That's or can both have something called a sporocarp. That's an interesting structure. So a sporocarp is a large, comparatively large, of course, for a fungus, structure that is going to rise above the substrate so that spores can rain out of it, fall out of it, disperse aerially out of it. So it's an aerial structure that rises up. mushrooms are a sporocarp, for instance. So both the ascomycetes and basidiomycetes can have sporocarps. So what's an ascomycete named after? It's named after the ascoma. That is the cup-shaped sporocarp that many ascomycetes have. And how's this for creativity? They also have asci, which are the actual structures that produce the spores. The actual sporangia themselves are called asci. And so they are named, so the spores themselves are called ascospores because they're produced in asci. So the ascoma and ascus are different things. So the ascoma is a structure that houses asci, and the asci are structures that produce the spores through, you guessed it, meiosis. Now the basidiomycetes, they are named after basidiomi. This is one you might have seen also as basidiocarp, and they tend to be club-shaped. So a mushroom, a bracket that you see, a puffball, those are all basidiomi, that you can see of basidiomycetes. So the basidioma is lined with basidia in the case of a mushroom, which is going to be most familiar. The gills are lined with basidia that produce basidiospores through meiosis. We'll see some examples of these. All three of these groups are kind of moldy for the most part. So if you see them growing in their substrate, they'll just look like mold, they'll look like threads, things like that. It's the sporocarp that you probably think of, though, when you see these things. So you might see the sporocarp of an ascomycete. You might see the sporocarp of a basidiomycete. Let's look at the life cycles of the ascomycetes and basidiomycetes. So the ascomycetes and basidiomycetes both are actually a little more derived compared to the zygomycetes. So all of these fungi have karyogamy as part of their sexual life cycle. So there are hyphae from different individuals that come together. We tend to say negative type, negative and positive. That is just a way to separate the different individuals. What's different, what's really derived about the ascomycetes and basidiomycetes, even though, which by the way are the majority of fungi, what's really derived is that the heterokaryotic stage is dikaryotic. So it's a little more controlled. So there are just two haploid nuclei in each cell. So whereas a zygomycete might have more than two haploid nuclei from multiple different cells all merging together, especially in the sporangium, the ascomycetes and basidiomycetes both are dikaryotic. So the dikaryotic cells will, at some point, the nuclei will merge. There will be zygote cells, and the zygote cells will actually go through meiosis. And when they go through meiosis, they produce, like in every example of meiosis, you should remember this from 1107, they'll produce 4 haploid daughter cells. What's unique about ascomycetes is that before the cells, these cells with 4 haploid nuclei divide, before that happens, the nuclei divide again. And so we actually end up with 8 spores per ascus. So 8 spores per cell. And so if you're looking at a fungus, if you're looking at a fruiting body, if you're looking at something you're trying to figure out if it's an ascus or basidium, count those spores. Look for those 8 daughters. So meiosis occurs and then mitosis occurs before anything divides. So we'll end up with 8 spores per ascus. Okay, spores will be dispersed from the cup-like ascocarp or ascioma, and then they will disperse into the environment and do their thing. Nice and haploid, of course. Okay, not too much to say about basidiomycetes. The life cycle itself is the same as ascomycetes. They are dikaryotic, just like the ascomycetes. Those are the only 2 fungi that are dikaryotic. But the major difference is going to be the sporocarp. So the basidioma, the basidiocarp, is going to have gills. Rather than being cup-shaped, it's going to be club-shaped. And those gills will be covered with the basidia themselves, which, just like the asci, they do look club-shaped. So how can you tell them apart? 4 nuclei in the basidium, which become 4 basidiospores. So again, when you're trying to tell the difference between a basidium and an ascus, look at the spores, or if the spores aren't formed yet, look at the nuclei. Look at those. Count. 4 versus 8. I'm looking into the future with my crystal ball. There's a pretty good chance on the lab exam you might have to be able to tell those apart. So make sure that you do that beforehand. The last group of fungi that I'll talk about are called the glomeromycetes. Glomero meaning ball, so they are going to have mycelium in the shape of balls. These actually are not the most derived fungi. So the most derived fungi are ascomycetes and basidiomycetes. The reason I'm covering these after the basidiomycetes and ascomycetes is because I want to talk about them mostly from the context of their interactions. So the glomeromycetes are interesting because they are symbionts, and they are symbionts of plants. In particular, they are symbionts of plant roots. And without them, the plants are in a world of hurt. So we have this term for a lot of the glomeromycetes. It's arbuscular mycorrhizal fungus. So let's kind of look up each of those terms. Arbuscular mycorrhizal fungi. So fungus is a pretty easy one. We don't have to worry about that. Arbuscular. What does arbuscular mean? So you've heard of Arbor Day. So again, same word root. So just tree-like, tree-shaped, tree-formed. Mycorrhizal. So hopefully after all this you've kind of got the idea that myco, M-Y-C, so if you see the M-Y-C-E-T word root or the M-Y-C-O word root, hopefully you'll recognize that word root means fungus. So mycorrhizal means, half of it means fungus. The rhizal means root. So they're tree-like, root, fungi. So arbuscular, mycorrhizal. Again, myco means fungus, rhizal means root. So what's amazing is that pretty much every plant that we find and actually look has some sort of mycorrhizae. And mycorrhizae are tremendously important to helping plants absorb nutrients. And so they help add more surface area. They might help break down things in ways the plant can't. They might make things more available to the plants. So things that, if you think about roots can't bring in very complex molecules, right? So the arbuscular mycorrhizal fungi are good at breaking them down. And so they can kind of grow either actually within or around roots. In this case here, in this picture from How Life Works, you can see some ectomycorrhizae, but there's also endomycorrhizae as well, which can grow into plant root cells. All right, so if you look at the diagram here, my nice Actor 1, Actor 2 table, if you think of these arbuscular mycorrhizal fungi in plants, which cell do you think that would fit in? So does the plant benefit? Is there a cost to the plant? Is there no effect on the plant? Does the fungus benefit? Does it have any is there any harm to the fungus? Is there does the fungus not get any outcome? So think about that. Why would they grow together if only one has a benefit or harm? Also think of that. It's really cool though. Pretty much every plant we look in has some sort of fungal symbiont. Some of them even have symbionts called endophytes, which are symbionts that don't necessarily have to be glomeromycetes, that can grow up into the leaves, into the shoot. Okay, let's ask the question here as we talk about a really cool group. Are you liking these creatures? So these are the lichens, so can't get enough of the good puns. So a lichen is an interesting creature. So it is considered a special organism with its own branch of study, lichenology. But a lichen is, and there's lichen taxonomists as well. But a lichen is interesting because it is exists as a symbiosis. It exists as a mutualism in particular, so a symbiosis in which both members of the symbiosis derive some benefit. So it can be pretty variable what type of creatures make up the lichen. There's hundreds of recognized lichen species, if not thousands. So the thing that they all have in common is there is an ascomycete fungus that is associating in some way with a small producer. So that small producer might be a chlorophyll green alga, so it might be a eukaryotic producer. It might be a cyanobacterium even, so it could be a prokaryotic producer. So essentially there has to be a fungus and a producer of some kind interacting. And as I'm sure you could all figure out, the benefits to both are very important to both of them. So the fungus is going to get a source of food by using the photosynthesis of the producer to the point that sometimes it can almost kind of veer into the parasitic relationship, and the producer is going to get a place to live. It's going to be able to live in a very hardy, and it's going to live in a very difficult environment to live in otherwise. So a producer that would ordinarily need to live in moist soil or a wetland can now live on rocks or on trees or in some other substrate like that. So lichens are really cool creatures. So they might look like crusts. They might look like dried paint. They might look really leafy, as you see in these foliose lichens on the right photo here. So they can look pretty cool. What I'll say is when you guys go to the woods, and some of the North Georgia woodlands are spectacular, especially after rain. Take a look at those lichens. You'll be pretty amazed by what you see. Maybe do some photography. Lichen taxonomy is actually pretty poorly known, and so if you go around and take some photos, you might discover a new species. It's a lot out there to be discovered, and they can live in what seem like outrageously harsh environments. So they can live, first of all, they can live on trees, which isn't a very easy way to get nutrients. They can live on rocks. They can live up north, so above the tree line, in the tundra. There's some of the few things out there. If you think about the tundra, they're the basis of a lot of those food webs. Okay, fungi can be antagonists. So by antagonists, I mean they derive some benefit out of some other organisms suffering. So here there's lots of examples. So you see here on the right there's two pictures, one of which is a slide mount, one of which is a scanning electron micrograph, two images of fungal hyphae growing within a host of some kind. The bottom right is the vascular tissue of a plant. So they can grow through something and be pretty nasty pathogens. We'll talk about some cool examples on the next slide. And then I also have this picture of the nematode being reeled in, basically being lassoed by a fungus and then being digested. So kind of an interesting thing to think about is fungi acting as predators and not just parasites and pathogens. So the pathogens aren't horrible problems to us if you think about it directly, right? So you're not too likely to die of athlete's foot. You're not too likely to die of a yeast infection. They're pretty awful to have, but they're not as likely to be lethal. However, that doesn't mean that we should discount how deadly fungal pathogens can be to animals. So I'll just bring up a couple examples here. So there's a chytrid called Batrachochytrium dendrobatidis that is a particularly nasty fungal pathogen, and it's one of the many causes of the amphibian decline. So globally, amphibians are going through a mass extinction right now, and it's pretty heartbreaking. And the chytrid fungus, which we generally just call BD because the scientific name is so hard to say, BD is wiping them out in some of these amphibian diversity hotspots. So it is in the southeastern U.S., South America. If you look at the rainforests of Australia, the edges of Australia, the South Pacific, some of these places with the most amphibians are being hit really hard by the BD pathogen. And it's a very notable and very important pathogen. And in fact, we have two faculty colleagues of mine. Dr. Hislop works with sampling amphibians to test for the BD pathogen, and Dr. Morgan takes those swabbed samples and tests them to see if they test positive or negative. So if anybody is interested in learning about some cool field research, they definitely are always interested in taking more students. Another really kind of infamous wildlife pathogen that's a fungus is called Geomyces destructans, and it's a gross ascomycete that causes white nose syndrome. And if you look at these poor bats, little brown bats, which we do have in Georgia, are some of the most threatened hosts. The Geomyces destructans hyphae actually grow out of the mucous membrane. So the hyphae grow through the whole body of the bat and cause a lot of destruction to the tissues and kill it. And the hyphae kind of grow out of to produce spores. They grow out of kind of the mucous membranes, in particular the nose. So you'll see these hyphae grow out of the nose, and so it's called a white nose syndrome. And they actually figured out, and they actually were able to culture the pathogen and do some interesting work. So they did Koch's postulates to figure out the causative agents of white nose syndrome. So yeah, they did isolate spores from diseased bats, grow them in a culture, and in fact, otherwise healthy bats. So they did this fairly recently. But white nose syndrome, I haven't heard much about it coming to Georgia, but it's a really big problem in the northeastern U.S., New England, and the upper mid-Atlantic states, New York, things like that, to the point that it's causing fungi and crops. So fungi and pathogens are a major problem to our crops. So I have a couple examples here of those. So I had to point out my very first job was I was the kind of culture, I don't know if you'd say specialist. I was the culture guy at NDSU for their Alternaria solani they're using, and it's a blight of tomatoes and potatoes. So it causes something called early blight, which is not to be confused with late blight, which is what caused the potato famine. And if you look at this image, sadly it wasn't mine. I never kept any pictures or really even took any good ones. Back in 1999, 2000, it wasn't as popular to take photos of stuff. But here's a picture of the spores of Alternaria solani. And so if you look at this and you see more than 4 of them, you should be able to recognize pretty quickly that this is an ascomycete fungus. So my very first job was to culture this little creature. In fact, I had to pick up individual spores to start the cultures on Petri plates. A couple other pathogens. By the way, that's a very important one, especially in areas where they grow potatoes as a large-scale crop. Rusts. Rusts of grains are arguably the most economically damaging pathogen around today. So rusts are fungi that look like rust, for lack of a better word. So they have brown, kind of fuzzy-looking conidia, and they tend to attack grain crops. And so there's a chance that some of these rusts can cause mass famine because we rely so much on grain now. If you thought the Irish relied on potatoes a lot back in history, imagine how much we rely on grain, and particularly wheat, as a civilization now. So if there's a massive wheat kill, we'll be in really big trouble. Okay, so let's talk a little bit about fungi. They can be pathogens, they can be mutualists. Let's talk a little bit about them as competitors, kind of as almost our last idea of fungi here. So fungi can compete with each other, and what's interesting is when organisms have multiple mutualists, they might not necessarily get along. So I'm sure some of you have friends that are friends with you, but they don't get along with each other, right? So here's a really interesting example that a scientist I have met worked on. So it turns out some of these mutualistic fungi that interact with plants might not get along with each other. So the arbuscular mycorrhizal fungi that live in the roots, and the endophyte fungi that live in the shoots, the leaves and stems, they don't really get along very well. And the endophytes function in a lot of ways to produce toxins, which we'll talk about more in just a second. They produce a lot of toxins to prevent herbivory, to keep the plant alive, and obviously themselves alive as well, since they grow in the plant. So they don't really get along. In fact, plants with endophytes have difficulty being colonized by AMFs, by the root fungi. And if the AMFs aren't colonizing, the plant won't grow as well. So there's this really interesting trade-off that the plant does better, or the plant does well with each of them, but together they just don't work out that well. So it's really a balancing act by how much does the plant need nutrients compared to how much does the plant need to be protected against herbivores. So that's really interesting. Fungi can compete with other living things. So it's really interesting to think about they can compete with things that use the same resources with them. So I'll give you a second to think of how fungi can compete. So let's say they are in a habitat. So they are going to grow into an organic substrate. So what other organisms might be growing into an organic substrate, sharing that organic substrate, using the same resources as fungi? Well, what comes to mind fastest for me, and hopefully for you also, will be bacteria. Well, how do fungi compete with bacteria? They can compete with bacteria through chemistry. So think about penicillin, right? So penicillin is going to be an antimicrobial agent. It's an antibiotic that's specific to gram-positive bacteria. And penicillin is produced by Penicillium. It's a fungus. One of many examples in which a chemical is named after the organism that produces it. Here's another example. And I don't know how many of you recognize the actual photograph, hopefully not too many of you, I'm sure not many of you recognize the chemical structure. This is psilocybin. Psilocybin is a molecule that is based on an amino acid derivative, and I'm sure many of you have heard, maybe somebody's tried, maybe some of you haven't, that this is a hallucinogen. It's an entheogen. It's going to create a religious experience, is what the term entheogen means. So we have all these kind of psychedelic looking pictures. We have an interesting experience when this molecule crosses the blood-brain barrier. So why might fungi have it? Obviously, fungi, or I won't say obviously, but it's very unlikely that they have it just to get us high, right? So it's there for another reason. So this and other chemicals that have interesting pharmacological effects tend to be there to prevent other microbes from attack, and these chemicals tend to be there in order to prevent herbivores or predators from eating the organism. So fungi use a lot of chemical warfare. So think about how many mushrooms you're not supposed to eat. So why aren't you supposed to eat most mushrooms? Because they're outrageously toxic and can even kill you. So fungi tend to evolve some pretty potent chemicals, whether they're antibiotics, like in the case of penicillium, whether they are have these crazy pharmacological effects like psilocybin, whether they are super toxic, like some of the things in some of the death cap mushrooms. Probably, arguably, the most interesting fungal compounds, by the way, even outside of psilocybin, are the ones that prevent alcohol metabolism. So some of these mushrooms that you eat, you can die very easily from drinking a very small amount of alcohol. All right, so that's defense. That's these chemicals can be used for defense against competitors, against predators, all those things. So let's finish off by looking at some interactions. So a couple more interactions between humans and animals, and I got some really nice electron micrographs out of Principles of Life, so we can look at each of these. So it turns out the fungus that produces blue cheese is very closely related to the fungus that we get penicillin from. And in fact, we literally get penicillin from mold, and we get cheese from mold. And what's interesting, if you think about the flavor of blue cheese, which I'm sure some of you love and some of you despise, you know, that flavor comes from the crazy compounds produced by these fungi. And these compounds are there to help digest the organic substrate to help out the fungus. And of course, the compounds are there to keep away potential bacterial competitors that might be in the cheese. So get some pretty interesting chemistry there. We also interact with yeasts, because we need yeast to make a lot of bread, not necessarily all bread, but the bread that we usually think of, yeast bread. And of course we need yeast to get the booze we drink. So this is kind of an interesting way to think about interactions, right? So we're basically feeding both of these fungi. We're feeding the penicillium-like fungus to make cheese. We're feeding the fungi that leaven our bread. We're feeding them dough. Hopefully your dough has honey or sugar in it or something that will make it rise better. We're feeding them dough. In the case of beer, we're feeding them malted barley. So it's really or sugar, by the way, if you hand bottle and carbonate. So it's really interesting to think about these interactions, because we definitely get a benefit out of these, right? We get cheese, we get beer, we get bread from fungi, and they get an interaction too, right? Or they get a benefit too, right? So they get a food source, they get a place to grow, they get help out with the reproduction. But what happens when we eat that cheese? We're probably eating the fungi alive. What happens when we bake that bread? These poor yeasts definitely don't survive that, right? what happens when you're fermenting your alcohol, when you're fermenting your wort. You are basically relying on them till they starve, and then you either let them starve or you just drink them alive, depending on what kind of a brewer you are. So, not being very nice in that case. So, it's interesting because it's a mutualistic interaction for a while, where then we kill them off. So, at some point, it becomes a negative interaction for them. So, interesting thing to think about. So, when it comes to the handout, this is a good idea for thought. Just make sure if you want to use an interaction like this, just make sure you carefully defend your point. Okay, so that completes my lecture on fungi. So what are some of the things that we want to keep in mind? What's left? So hopefully you recognize that a fungus is an organism that is a lot like us, but they don't have a body in the same way we do. We'll cover that more with animals. So they're very close relatives of us, but they live by growing in organic substrates and digesting that organic substrate outside of the organism and then absorbing the nutrients that they've digested externally. So fungus is a lot like us, except the way it grows. And fungi are also very different from us in the way they reproduce. In particular, the fact that most fungi have this weird heterokaryotic life stage that animals don't have. So with that, that's kind of fungi in a nutshell. Lots of interactions, lots of cool diversity, lots of cool life cycles. So with that, thank you and good luck on your lecture quiz.